Can you answer this biology question? It's about DNA and something called gel electrophoresis. These are strands of DNA, and each of the strands is a different length. If the samples of DNA were electrophoresed through an agarose gel, which fragment of DNA is the arrow pointing to? So if we take a look at our DNA, we can see that the arrow is pointing to this strand. You can see our gel plate here with our ladder in lane 1 and our fragments in lane 2. Each of these fragments is labeled with a letter A through E. So which letter is corresponding to the fragment that the arrow is pointing to? Remember that the gel is going to separate DNA based on length. Shorter fragments are going to go the furthest, while the longer fragments don't travel as far. So you can see here, E is the shortest fragment, while A is the largest fragment. If we look at our DNA fragments, we can see that the arrow is pointing to the fragment that's the second longest. And you can see in lane 2, B is going to be the second longest fragment. So the answer to our question is that this band is representing the fragment that the arrow is pointing to. Now let's go a step further. Around how large is fragment B? All you have to do to figure this question out is figure out where this band is based on our size ladder. You can see that band B is between the 8 and 6 band, which means it's going to be 7 kilobases long.